Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Hi, welcome everybody back to In the Interim. I'm your host, Scott Berry, and I have a distinguished guest today. I have Jeff Saver, Dr. Jeff Saver, who is the Carol and James Collins Distinguished Professor of Neurology at the David Geffen School of Medicine, UCLA. He is the director of the UCLA Comprehensive Stroke and Vascular Neurology program. And I'll talk more about his distinguished history as we go, but welcome to In the Interim, Jeff.
Thank you, Scott. It's such a pleasure to be here with you and your audience.
Wonderful. So we're going to touch on a number of topics, but first let's talk about now you're a distinguished clinical trialist. You've worked a great deal in stroke, quantifying the effects of stroke, things like PFO occlusion, which I may ask about that as well. But a good number of things in stroke and the clinical trials. But let's talk a little bit about how you got here within this. So you went to Harvard as an undergrad. Interestingly, I think you were a philosophy and biomedical sort of double major. When you went to Harvard as an undergrad, was it always that you were going to be a doctor? Was that the plan?
Well, I do come from a medical family. My grandfather and father were medical physicians, so there was a strong push to go into medicine, against which I, of course, rebelled somewhat. And I originally was going to secretly triumph as a basic scientist with an MD degree, but after spending a summer in the basic science lab deprived of interaction with people, I realized that wasn't for me. And I was tempted by philosophy. I actually applied for it and received a fellowship from Harvard in the year after that. Was originally to give in the 1890s Harvard gentlemen grand tours of Europe. Of course, the Harvard faculty could no longer allow you to just travel, so you had to have a project. So my project was to interview biologists on religion and biology in four countries: England, France, Israel, and Japan. Went around the world, got some need to be out there out of my system, and then went to medical school somewhat reluctantly, but did go there. And I knew once I arrived it was either neurology or psychiatry. Nothing else, had that deep connection to the mind and who we are as humans. So that's how I got started.
Was your family, or your father, your grandfather, were they in neurology, neuroscience, in their medicine?
No, they were. My father was an old-fashioned GP in Boston. When I was a toddler, it drove me around on his home calls. And my dad was an internist in the New York City hospital system.
Okay. So you grew up in New England, hence going to Harvard.
Well, I grew up in New York, but Northeast.
Okay.
Absolutely.
Okay. And from there, you go to medical school, and you're vascular neurology, and you do a number of programs in that. At what point did clinical trials become interesting to you?
Well, I originally set out to be a cognitive and behavioral neurologist, and that was my first post-training fellowship at the University of Iowa with the Damasio's. But it was intellectually fascinating, but we couldn't do anything for patients. And in Iowa was the very first stroke unit in the country, Harold Adams and Jose Miller, doing one of the very first big multicenter trials in the country, the TOAST trial of an acute anticoagulant. And so as I was having my disappointments as well as encouragements in neurobehavior, I saw this alternative approach, which was much more collaborative, much more therapeutically oriented, much more engaged directly with patients on their benefit. So that's why I then went on to immediately do a stroke fellowship at Brown and on to the career, including clinical trials. I will say, when I set out, part of my goal was, reason for going to clinical trials of stroke was I didn't anticipate to ever be writing grants, living on grants. And in clinical trials, you could be a site investigator, contribute to big trials, and be a cog in a machine and be very helpful. And that made it very attractive to me. I was a late bloomer. I didn't write my first grant until late assistant professorhood. But what happened as I was doing clinical trials was I always wanted to understand more about what we were doing. And neurology is, if people talk a lot about burnout in medicine, but I think for lots of medicine, and especially for neurology, a big problem is burn-in. It takes such a long time to acquire the huge knowledge base of neurologic disease. You don't know if you're going to be good until you're in your late 30s, early 40s. And I had a long burn-in period, but it was very rewarding along the long apprenticeship. And then I eventually took on more and more leadership roles and ended up writing grants in the way I never expected to.
So, well, by the way, what was the result of the TOAST trial?
The TOAST trial was neutral. It was done at the same time as the NINS TPA trials. They were the two great strides. But the TOAST trial set the framework for all trials that followed. We still use the TOAST system for classifying the cause of stroke now 30 years later.
Okay, so you're jumping into stroke, which you do a lot of work within stroke. Early on, by the way, I was looking at your various publications, and it's hard to count your publications, but 870 at one source, 793 in another source. But one of the most cited of those is called Time is Brain Quantified, and it talks about stroke. So you get involved in a great deal, and we're going to sort of touch on stroke, and we're going to touch on how to analyze stroke trials, but acute stroke. So I think given that paper and Time is Brain, what is an acute stroke and what happens to the brain during an acute stroke?
Sure. Well, there are two main types of stroke. Stroke is injury to the brain from blockages or ruptures of blood vessels. And a blockage in a blood vessel, usually due to a clot, is the cause of 4 out of 5 strokes. Rupture of a blood vessel causing a bleeding in the brain, a hemorrhagic stroke, 1 out of 5. The blockage stroke, ischemic stroke, when the blockage occurs, a small group of nerve cells is deprived of all blood flow, all nutrients, and dies within a few minutes. But a much larger group of nerve cells has a more moderate reduction in blood flow because there are some collateral channels still delivering blood, and the tissue there can last for a few hours to up to 24 hours, depending on each patient's anatomy. So that is the target of acute ischemic stroke therapy, getting the vessel open and restoring blood flow in time to save those threatened but still salvageable brain tissues.
Okay, so as in my naive statistician view of this, within the brain, blood flows through the brain and it delivers oxygen. I'm sure it does much more than that. But when a clot occurs, you don't get the blood flow. The downstream parts of the brain that are served by that are no longer served. There's, as you say, 1.9 million neurons, 14 billion synapses, and 7 miles of myelinated fibers are destroyed by the not having blood flow. presumably,
minute.
and you get this
Yes.
death every minute,
2 million
yes.
nerve cells a minute. And there's an interesting clinical trial story behind my doing that article to quantify the speed of loss of neural circuitry in the brain. When we were doing acute stroke trials in the late 90s, early 2000s, the IRB would ask us to make sure that we had an informed consent, and they wanted us to wait an hour to give patients and families time to think about it before signing the consent form, which is absolutely appropriate to maximize the ethical consideration by the patient. But the problem is the brain is dying all that time, and your chance to show benefit of drug is being lost, and also the patient's ability to themselves experience benefit from the intervention is being lost. And I couldn't make that clear to the IRBs. I said, you can't delay it. And they said, well, we're going to wait an hour. So I said, all right, I'm going to look at the literature, and there was some interesting stereotaxic literature on just how many 1 billion brain cells there are in the brain and the typical size of an ischemic stroke and how long it takes to grow. And from that, you could derive that it's 2 million nerve cells lost a minute. And the rallying cry of time is brain had already existed, but being able to quantify it gave a big kick to that, both in research but also in clinical care.
So, I mean, the analogy would be sort of somebody's drowning, and you wait for a period of time to enroll them in a trial rather than doing something to fix the drowning. Okay. So stroke has had an amazing, and your involvement in trials has an amazing history, and endovascular therapy, which you can describe this, but you go in and you remove the clot, restoring blood flow, has been amazingly successful through a history of sort of figuring out the right patients and the wrong patients and the right devices and all of this. But this incredible device that saves many, many lives in acute stroke, you were involved in the beginning of the development of this device. Sounds like an incredible story.
Yeah, it was a great privilege. I had the great fortune of being at UCLA, where a French interventional neurology, Pierre Gobin, invented the very first retrieval devices. These are devices where the interventionalist will put a wire and small tube into an artery in the leg and slowly advance it up into the brain, reach the clot, and then either deploy a grabbing tool to pull the clot out or suction to pull the clot out and reopen the artery and restore blood flow. Pierre, his design was of a helical coil that he would put into the clot and then pull it out, much like a corkscrew pulling cork out of a wine bottle. It is not an accident that a French interventional neurologist developed this particular device as the first device. And he did the very first animal models, and then when it came time to go to humans, I, as a non-invasive stroke neurologist, helped to design the very first studies and was able to be involved ever since.
So the clot, is this what we think of as, I mean, what is it you're removing? Is this coagulated blood that doesn't flow anymore? Is that what the clot is?
Yes, and it can have a variety of compositions. It can be mostly red blood cells. It can be mostly platelet clumping elements of the blood. It's always a combination of the two. But the same clots that we see on our skin when we have a skin break and we see clots form, this is what forms. But instead of forming in a useful way to heal a broken surface, it forms within the lumen of the artery, or travels to, forms elsewhere and travels there and completely blocks that artery blood flow.
Amazing. And just how long does it take when you start this procedure to remove the clot? You described time as brain. How long would it take for this to happen?
From the time that the skin puncture occurs to begin the procedure. These days, they can reach the clot within 25 minutes, often be able to pull it out with the first pass within 35 minutes. When we first started doing this, we got the artery fully open maybe 25% of the time. And then over time, there's been a history of incremental improvements in the devices. Now, the interventionalists get the artery open 85, 90% of the time.
Wow, that's amazing. It's incredible the story of this and the impact it's had. So now we're involved. You're involved as a investigator, primary investigator in the STEP platform, which is now looking. You've identified people that absolutely benefit by this endovascular therapy based on the location, the size of the stroke, the time since the stroke started. But the question is, who does this benefit? So a number of clinical trials, a number of stroke clinical trials, and there's been a history of endpoints in stroke trials. It seems like, there was a Scandinavian stroke scale, there was other things, but at least it has come to the endpoint being the modified Rankin score, which is an ordinal score, is measuring the neurological status of a patient. And I've talked about this before. Maybe we should talk about that scale. But then how do you analyze this ordinal scale? And we'll sort of come to that. So to tell our audience what the MRS ordinal scale is.
Sure. So it's now a 7-level scale going from the best 0, recovered to no symptoms whatsoever, to the worst 6, dead. And in between are different steps. 1 is you have symptoms, but you can work. 2 is you can't work, but you can live alone. 3 is you can't live alone, but you can walk. 4 is you can't walk, and a 5 is you're bedridden, need somebody with you all the time. So each has a pretty clear step down, although just how much worse it is to be able to have symptoms versus no symptoms versus being able to walk versus not able to walk, the value of those steps are unlikely to be exactly equal. So it makes this an ranked but ordinal scale where we don't know the distance between each level.
And the last level on this is death. So from the bedridden, then there is a value that is death.
Yes.
Within that. So you touched on exactly the issue of this. As a statistician, if we do something like we call it the shift analysis or a proportional odds model, we're analyzing this that 0 is the best, 1 is next, 2 is next, but there's no numerical difference between them. We don't say 1 is a little bit better than 0, more than 2 is better than 1, and likewise. We don't actually talk about the utility of death relative to other scales. It leads to some challenges in the interpretation of outcomes if we rely on a statistical assumption to dictate what is good clinical outcomes. So you were involved, and I was involved in an effort to develop a different way to analyze this. So what was an alternative way to analyze it?
So we were part of the group that developed the utility-weighted modified Rankin scale, the UWMRS, which assigns a health utility value to each level of the 7 levels. Weight between 0 and 1.0, 1.0 being fully healthy, vigorous, 0 being dead, and this is the standard way to measure the value of health states in health econometrics. So what were the value of each Rankin scale? We took 2 approaches to figure that out. One was John Rothwell and the Oxford group in Britain had done a large study in thousands of stroke survivors, scoring them on the Rankin scale and having them themselves fill out the European Quality of Life scale for their health-related quality of life. And so they were able to derive from patients at each level what the average value of the patients was. Our group used the World Health Organization Global Burden of Disease approach, where they have to assign the value of every disease known to humans. And the way they do that is with the time trade-off, person trade-off measure, that if you were a physician making health system decisions, and you could keep either 1,000 patients who are completely healthy alive for another year, or X number of patients with whatever disease state they have alive for a year, how many patients with that disease state would you need to be indifferent as to where you spent the money? And if it's a nearly completely healthy, it's not going to be that many more patients. If they're really bad, it's going to be a lot more patients. So we did that with stroke physicians and nurses from around the world and came up with the clinician ratings. And we looked at the two to take the average between the patient self-ratings and the clinician ratings, and it turned out they were remarkably similar. They matched almost perfectly, which was very reassuring. And we came up with these weights for each of the 7 levels.
As an example, in the numeric values, you describe this 0 to 1. So 0 is perfect neurological status. 1, you described it, there's some deficit, but you're fully working, fully independent. That is a 1 for the value 0 and 0.91 for the value 1. So there's a 0.09 lost going from 0 to 1. Likewise, on the far end of the scale, 5 and 6 are actually combined. So being bedridden, essentially vegetative and death are combined to have 0. And stepping up to 4 is 0.33 units. So going from that 5 to 4 is 0.33 units. We're going from 1 to 0 is 0.09 units. So it's largely 4 times as good to move patients from 5, 6 to 4 that it is from 1 to 0. And that goes into the analysis of the endpoint at the end of the day. And likewise, there are values across the scale of these to look at. So this is fantastic, and I love it for multiple reasons. In some trials, and especially in stroke, like endovascular therapy, you worried you might actually be doing harm to some patients. Maybe by restoring blood flow, there isn't much brain to save, and you actually cause bleeding, you cause harm. And if we do an analysis and say, well, we're looking at efficacy, but now how do we incorporate safety? This incorporates it all. It incorporates the negative outcomes of this. This endpoint encompasses that, and it weights the various values. So I love this. I think it works great. What has been the reaction from the stroke community to the development of this endpoint?
Well, there have been a range of reactions. Some people have really loved it, adopted it, and been very happy about it. Others have, the regulators at FDA and elsewhere across the world have been hesitant. They've already been hesitant about looking at the Rankin Scale as an ordinal outcome. They've preferred to dichotomize the Rankin Scale and say, well, analyze 0 to 2 versus 3 to 6, because that's computationally easier, but also because then it's easy to derive a number needed to treat so clinicians can understand what they're doing. Now, of course, this is a completely mistaken idea, because if you just count improvements in the health state transition across one level of the Rankin, you are missing improvements elsewhere. You're giving misleading clinicians as to the actual benefit of your therapy. And the example for lay people I give is if you had a new high school educational intervention and you wanted to see if it helped students, would you only count how many people went from a B to a B plus, or would you want to count everybody? So we've had those difficulties with the regulators. And then there other folks who think appropriately that the Rankin utility weights that we derived are based on originally patients from Great Britain plus this international panel. And every country has its own tariffs for utility weights, and they value different outcomes. Rankin 1 in India is very different than a Rankin 1 in the US. And that's certainly true. But what's also true is that counting every level of the Rankin equally as one is always further off from what people actually value than comparing one country to another country. That's always much closer.
Yeah. And the dichotomous outcome, for example, you could write down a utility function, which is 1, 1, 1, 0, 0, 0, 0, and say that, let's use that utility function, which is one that nobody has in that, rather than the one that you developed that represents a population. Alternatively, even doing an ordinal analysis imposes a utility upon these states, but in an unspecified, implicit way that comes through the proportionality assumption. So you are still assuming that everybody in the population has a particular utility that's just implicit in the statistical algorithm, which some people seem sort of more comfortable with than explicitly writing it down.
Yeah, it's important to emphasize that you can't escape putting weights on each level of the Rankin, no matter how you analyze. If you dichotomize, as you say, you're weighting many of them as worthless. If you analyze ordinally, then you're implicitly ranking them in somewhat obscure but relatively equal ways. And if you do it using open utility weights that are empirically derived, you're basing it on real patients and real data.
And there was the Dawn trial used utility-weighted MRS, and Mr. Clean did not. And the Mr. Clean investigators had a series that showed up where they complained about the utility weight, and it's a fascinating read of their response to it, our response back to them, their response on the various methods. So this is something that is passionate about how to analyze this endpoint with disagreement, for sure.
Yes, I just had an exchange 3 days ago about a new analytic project with the Mr. Clean investigators, where they, again, were objecting to the utility-weighted MRS. So this controversy has not died down, but I think we are slowly winning the field.
So this interesting question you brought up about the number needed to treat on a dichotomous endpoint is really simple. You've done work on number needed to treat for this utility weighted, if that's the thing that is relevant to clinicians.
Yes, and we've done a lot of work on ordinal derivation of utility to treat, and there are a variety of techniques that we have been developed. Our groups used a variety of joint outcome table specification techniques, and whatever you do, you're estimating the number needed to treat, because parallel group trials, unlike crossover trials, won't let you specify it exactly when you have an ordinal outcome, whereas you will exactly calculate it with dichotomous. But there's a famous quote that it is preferable to have an estimate of a useful outcome than an exact quantification of a not useful outcome, and I think we need to recognize that. And then the utility-weighted MRS then allows us to convert the value of these outcomes to health utility and the number needed to treat and the economic applications that are used worldwide in health system planning.
So it's and so this will continue. We will continue to push regulators. We'll continue to push better ways to analyze this. Interestingly, so it seems like a lot of the stroke world, the work so far, and this incredible work in a clot clearing device and medical thrombolytics seem to be the same mechanism of action of restoring blood flow. I know there's this huge interest in neuroprotectants. And I guess the idea behind a neuroprotectant is a lot of this research so far is clearing the clot, restoring the blood flow, letting the human body do the work it's supposed to. And you talked about synapses loss, all the things that dies during this process. Is a neuroprotectant something that we could give somebody while this is happening that might protect the synapses, protect the neurons, protect the myelinated fiber so it doesn't die? Is that the notion of a neuroprotectant?
Yes, the neuroprotective agents are agents that allow brain tissues to tolerate low blood flow, either while they're still not getting blood flow so they can last longer until you can restore the blood flow, or after the blood flow's been restored when you get reperfusion injury to protect them against that. And it turns out that plumbing is easy. We use lytic drugs like Drano. We use these devices like Roto-Rooter, and whereas intermediate metabolism of the nervous system is complicated, and so we've tried hundreds of neuroprotective drugs that worked in animals. None have worked so far in humans, but we need to get there because by the time we get the artery open with the thrombectomy devices, a lot of injury has often occurred. So being able to give a drug right at the onset or even before the onset that will protect the brain and deliver more salvageable tissue to the interventionalist is a critical need.
So for somebody who's spent so much time on the details of the brain, of what a clot is, the damage that happens, are you optimistic we'll find a neuroprotectant?
I'm sure we'll get there eventually. And there's actually a stream of trials coming out of China now where they are able to test large populations much more quickly than we've been able in the U.S. that are finally beginning to suggest positive results with neuroprotectants. And we in the NIH STEP group also have neuroprotective agents, including the uric acid that went through rigorous risk qualification in preclinical studies at NIH that we're very hopeful about. So I think in the next 5 years, we will have a neuroprotectant. Now, I will note this is my 30th year of saying in the next 5 years we'll have a neuroprotectant, but I am more hopeful than ever.
Okay. I assume, by the way, that humans have different capability to withstand lack of blood flow for a while, that there's natural heterogeneity and there may be protein levels and other things that just allow one person does much better where another does much worse?
We're not all that different in our ability to withstand low blood flow if exposed to the same amount of low blood flow. If we have a cardiac arrest, just about everybody only has 6 minutes at most.
Okay.
But we differ a lot in when there's a blockage in one artery, how much the other arteries can take over. So that makes for a very heterogeneous pattern. Some people are fast progressors with their stroke. Some people are slow progressors with their stroke, but it's due to the plumbing more than the neurochemistry.
And you've done cognitive behavioral work, vascular specialist and I know Alzheimer's is another disease where we talk about amyloid, plaque, and tau tangles kind of blocking the networking, and that's the loss of memory, the loss of cognition. Is that similar to a stroke, or are they just completely different blockage mechanisms vascularly?
They are different. Those are buildup of precipitated proteins in nerve cells, blocking the function of the nerve cells. as opposed to the clots that are in the arteries blocking the passage of flow through the arteries. But it is the case that most patients with dementia have mixed Alzheimer's disease and vascular dementia. That lifelong mild low blood flow injury to the brain adds to the Alzheimer's disease. In fact, the age-adjusted incidence of dementia now is half what it was 30 years ago. And that's because we're so much better at preventing vascular disease. We don't realize that because the population's been getting older, so more and more people are getting Alzheimer's. The prevalence is increasing, but if we hadn't reduced the age-adjusted incidence, it would have been even worse. So it's an untold success, unrecognized success story that we've cut the incidence of dementia in half, but of course we need to do more.
So what are those vascular things? Is it metabolic? Is it obesity? Cardiovascular drugs? What is it that we're doing better vascularly?
It is people have stopped smoking, people exercise more, people are on cholesterol-lowering medicines, we have better blood pressure-lowering medicines. All of those have added together.
Wow. Wow, it's amazing. Amazing work. Very interesting stuff. Can't wait to see more of these trials in neuroprotectants using the utility-weighted MRS, hopefully as we go forward. So, Jeff, I want to thank you tremendously for being on here. It's incredibly interesting. I love this stuff. So thank you for joining us here in the Interim.
Thank you so much. Thank you for helping our field so much, and thank you for having me, Scott.
Appreciate it. Thanks, Jeff.